So I'm very pleased today to present the Grand Round speaker, Dr. Jason Locasale, the Assistant Professor in the Department of Pharmacology and Cancer Biology at Duke University. Jason received his BA from Rutgers University in 2003, his PhD in 2008 from the Department of Biological Engineering at MIT. Actually, he started his graduate studies at UC Berkeley and then moved with his advisor, Arup Chakrabarty. Dr. Arup Chakrabarty to MIT, where he graduated with a PhD in 2008. His graduate training was broadly spanned physical chemistry, computational biology, immunology, and signal transduction. He did his postdoctoral training in Luke Cantley's lab at Harvard Medical School, where he developed an interest in cancer metabolism. Then he worked at Harvard Medical School for a year as an instructor. Then he spent 3 years as an assistant professor at Cornell before he accepted a position at the Department of Pharmacology and Cancer Biology at Duke University in 2015, and he's been there since then. Jason's graduate training in the physical sciences and in computational biology, coupled with his postdoctoral experience in cancer and metabolism, has allowed him to make meaningful contributions in a wide range of research areas. He authored a long list of diverse, high-impact publications during his years as a graduate student and postdoc. He currently heads an equally diverse research group at Duke and continues to publish in high-impact journals such as Cell Metabolism, Nature Cell Biology, Cell Reports at a very impressive rate. His lab applies state-of-the-art metabolomics technologies to study aspects of cancer biology, nutrition, molecular physiology. He's directed some of his recent efforts towards understanding the role of altered metabolism in cancer, in its effects on cellular physiology and its influence on the chromatin state. Focusing in particular on one-carbon metabolism, the methionine cycle, and histone methylation. Today, the role of reprogrammed metabolic pathways in altering the epigenetic landscape is being increasingly recognized, and the Locasale lab is contributing in no small way to a better understanding of this form of cellular regulation in the context of cancer in particular. I thought he was going to tell us all about it in his talk on cancer metabolism and epigenetics, but he tells me he's kind of changed his mind. So it's going to be more cancer metabolism and less epigenetics. So I'm sorry, guys. It'll be good anyway. So Dr. Locasale, the floor is yours.
Okay, thanks, Anita, for a nice introduction. Yeah, it's great to be here. It's actually my first time in Minnesota, and yeah, I mean, we've had like record cold in North Carolina this year, so it feels like home. Yeah, it's actually my third winter there. This is the first year I've had to wear winter clothing, and I have to worry about a snowstorm coming, which is almost unheard of in that area. Yeah, so it's great to be here. Yes, I titled the talk Cancer Metabolism and Epigenetics, and I was telling Amita yesterday, I was looking through it past couple days. I realized it was perhaps a little too long, so I took out a little bit of the epigenetics, but there's still plenty of things to talk about there. Okay, so just to kind of give a basic overview and what we work on, and that's metabolism, and what we study, and the basic ideas of how metabolism functions. And so basically, this is one of the key pathways we work on, known as central carbon metabolism, where the basic idea is that you have nutrients from the environment, and they're taken up, and there's a lot of complicated biochemistry that happens through which these nutrients are chemically modified and converted into other molecules. And as a result of this, and these networks are fairly elaborate, and they spread throughout thousands of chemical reactions. But the basic principles are that when these nutrients come in, they go through these processing centers, like for instance this this electron transport chain or TCA cycle shown here, and then they output series of biological functions that confer specific fitness advantages to cells. For instance, like these nutrients come in the form of glucose, lipids, amino acids into this mitochondria. They produce energy. They produce materials to regenerate or produce new cells. They also provide, through which this chemistry happens, they also provide intermediates that then couple to other molecular pathways downstream of metabolism, and that essentially is a form of signal transduction. For instance, reactive oxygen species are generated in this mitochondria. These reactive oxygen species, for instance, interact with protein phosphatases, and then signal transduction cascades are initiated through those events. And then something I'll talk about later is that, another thing we're learning about how metabolism is wired and is that it also confers ways to have direct communication to what we call epigenetics, or basically this is how histones are modified, and I'll explain some of the principles of that. So this is just another example of one of these pathways. This is known as one-carbon metabolism. The idea is the same, as that nutrients from the environment, and in the case of the central carbon metabolism, it was the macronutrients like lipids, like carbohydrates, proteins, fats. In this case, it's there's a series of micronutrients, things like involving vitamins, but also amino acids that come into this metabolic network. They get processed, and this is a simplification of a set of hundreds of chemical reactions, and then functions essentially are conferred to different aspects of cellular physiology. For instance, in this case of one-carbon metabolism, these nutrients come in, they get processed, and then the outputs are things like the synthesis of nucleotides, the maintenance of cellular redox potential, and also the name one-carbon metabolism. These nutrients, a single carbon moves around these cycles and is output in the form of a methyl group that's used for something like modifying histones in DNA. So in the context of cancer, there's been a lot of attention, especially over the last 10 years or so, into thinking about how we can understand these networks and these pathways in order to thinking about therapeutic aspects of cancer. And these are essentially four directions that cancer metabolism has either made or is making an impact in clinical situations. One is in the case of cancer, some of the most commonly used, classically known chemotherapeutic agents actually target metabolism, things like methotrexate, 5-FU, pemetrexed, gemcitabine, so on. And these, they are agents that produce pretty substantial clinical responses in patients. And so I think in terms of thinking about things like, for instance, that one-carbon cycle, there's a lot of effort these days in terms of understanding at the molecular level how some of these agents work. And so conventionally they thought to have been non-specific agents that that uniformly inhibit proliferating cells, but now we're learning that there's a lot of context and specificity associated with how these pathways are working and when these agents that target these drugs may be effective. Another aspect of cancer metabolism that is a major effort is that we already have a lot of drugs that are used for other indications that target metabolism, and there's certainly a lot of effort in understanding how and whether, in any case, these drugs could be used to affect outcomes in cancer. I guess the most common example is this drug metformin. It's in a clinical context, these are drugs that need no introduction, but metformin is the most commonly used agent in type 2 diabetes, and there's been a lot of epidemiology and even as far as phase 3 trials in patients right now that have shown that in some instances metformin actually has clinical benefits. And then the other thing with cancer metabolism is that these are metabolic enzymes, and so as a result of being an enzyme, these enzymes are evolved to bind to and react with and convert small molecules, and so they're naturally thought to be druggable either through the catalytic sites of these enzymes or through allosteric interactions that these enzymes have evolved. And something I'll talk about towards the end of the talk is that in addition to these things, we're starting to think about these concepts in cancer metabolism in ways that basic either lifestyle or specific interventions from environmental factors can actually be used to influence cancer, things like diet and exercise. And so I'll talk about some clinical work we're doing, later in the talk, where you could actually consider specific diets to affect, and then we consider the metabolism of patients, that these diets can actually affect the metabolism of patients in similar ways in which targeting, for instance, some of these pathways that I was just talking about could be achieved. So our lab's really focused on 3 directions, 3 basic areas, and they're all kind of coupled together, but one is, and I'll introduce this in a little bit, is that we develop and apply a lot of what we call metabolomics technology, and we use it to study the quantitative aspects of how metabolic pathways function. We also study how metabolism interacts with chromatin state and epigenetics, and I'll discuss that a little bit as well. And then the other thing we work on are translational aspects of understanding how metabolic pathways function in the ways that it can be affected in cancer settings through many of those mechanisms which I briefly introduced. Okay, so overview of the talk. As I mentioned, I'll first introduce briefly some of the metabolomics methodologies that our lab has been developing and applying, and then I'll talk about some of our efforts, particularly to target this phenomenon known as the Warburg effect in the context of thinking about aberrant and altered glucose metabolism in cancer, and then I'll talk about one-carbon metabolism, particularly how it's intersecting with chromatin biology and epigenetics, and ways in which diet and specific ways in which nutritional aspects of ways in which nutrition can influence the metabolic flow of the pathway in ways to affect specific and controllable biological outcomes. Okay, so metabolomics. The basic technology we've been developing over the years essentially is an approach involving chromatography coupled to mass spectrometry, where the basic idea is that an extract is prepared in cells, tissues, biological fluids. It's separated via an HPLC. It's injected into an ion source, and I guess, long story short, there's a series of ion optics where eventually the ions oscillate, and the frequency of oscillations tells you something about their mass. These things are recorded, and then there's algorithms that are applied to the data. And then, I guess, long story short, is in about a 30-minute experiment, we measure about 400 or so metabolites that we can identify in a targeted way, and we can also potentially identify something on the order of 10,000 with lesser confidence. This is just an example of what a data set looks like. This is 20 microliters of human plasma or serum, actually, across a cohort of 125 subjects, and this is just grouping these metabolites in different pathways, and you can see that there's a lot of data in this. And the other thing we do is we introduce isotopically labeled nutrients, either in cell culture or in the diets of animals or in the injection of into different aspects of circulation, and these isotopically labeled nutrients then spread throughout these metabolic pathways, and then from how they spread throughout the metabolic pathways, metabolic fluxes, or how things are flowing from one point to another in the pathway, can be inferred. And we do this with these mathematical models where basically we have how these nutrients are distributed across the pathways, and then what does that say about how things are flowing, and there's some chemical kinetic equations. Actually, University of Minnesota is one of the strongest places in the world for this sort of stuff. The chemical engineering department's very strong here. It's one of the best, like Minnesota and MIT are known as the two best chemical engineering. But okay, so Warburg effect. Okay, so just to introduce the basic, and this is something that probably doesn't need a lot of introduction at this point, but the basic idea is that it's one of the first molecularly noted differences between normal and tumor cells, and the basic observation that was made close to 100 years ago was that tumor tissue was taking up a lot of glucose, and by a lot of glucose, Warburg was actually doing a lot of quantitative biochemistry and physiology at the time, and basically the kind of numbers he was finding was that the amount of glucose coming into tumors was comparable, if not larger, than the amount of glucose coming into entire regions of the circulatory system. Like the amount of glucose coming in the tumor was like more, like a small tumor was more than what was coming into like all of muscle, for instance. And this was occurring independent of the presence or absence of oxygen, because it was known at the time that that this glycolysis, or this uptake of this glucose, was essentially regulated by oxygen in many physiological systems. But he found in tumors that that this glucose uptake was occurring independent of oxygen availability. And furthermore, in addition to this massive amount of glucose coming in, it was being fermented. So in the same way unicellular organisms like bacteria and yeast ferment glucose, ferment sugar, and make things like alcohol, tumors were fermenting the sugar, making lactic acid. And so basically this is the Warburg effect phenotype, is that you have massive amounts of glucose uptake, and then rather than this glucose being entered into the mitochondria, it is fermented as lactate. And then just many years later, this is something that's commonly considered in the case of in cancer diagnosis. For instance, we all know FDG PET is one way in which tumors are imaged and localized. And basically FDG is a radioactive version of glucose that is taken up but not further metabolized, and so the signal essentially is a measure of the rate of glucose uptake. So, so biology of the Warburg effect. Turns out it's actually fairly complex, and it's something that's been, I guess, extensively studied, especially over the last 10 years or so, where, I guess, where we start, I guess, about 10 years or so, it was starting to be appreciated that pretty much every oncogene and tumor suppressor gene that we knew of had an immediate and direct effect on regulating this Warburg effect, and it was something that was reversibly controlled by oncogenes, and also there were other experiments coming out at the time where it was at least shown in some genetic contexts that the Warburg effect was essentially required, or at least glucose metabolism. Tumor cells couldn't grow without glucose. Okay, so then what's the function, or what kind of advantage does this metabolic phenotype confer to cancer cells? And that's actually been fairly controversial, and and I'll just go through some of the basic hypotheses that have been proposed. One is that it's simply a kinetic effect, is that it's something where it takes about a minute or so to make lactic acid from glucose and make ATP that way, whereas it takes about a half hour to go into the mitochondria and get fully oxidized and have all the electrons get through and all the oxidative phosphorylation to happen. And so in cases where, for instance, there are two cells competing for a limited amount of glucose, the one that has a faster way of metabolizing it typically wins. Another possibility or hypothesis is that this is some phenomenon of a multicellular, it's another phenomenon of a multicellular environment where fermenting the glucose provides advantages to cancer cells relative to cells in the environment. For instance, some of the ideas there is that you make the lactic acid keeps the immune system away, and there's been some papers showing that lactic acid can polarize macrophages, for instance, and convert them into a repressive state. Another idea there is that the Warburg effect the pH decrease can allow for breakdown of tissue and so on. Another proposal, and this was largely proposed by Craig Thompson maybe about 15 or so years ago, is that this Warburg effect was an adaptation to support the proliferative demands of cancer cells. That cancer cells needed to figure out ways to take up the glucose, metabolize the glucose, and convert it to anabolic products, building blocks essentially, that allow for the production of new cells. So the glucose comes in, it makes lipids, it makes nucleotides, it makes protein, and so this Warburg effect is an adaptation to support that process. Another idea is that it may confer direct signaling functions to cells, and I'll show a little bit about how that might happen in a bit. Is that essentially this Warburg effect then allows for some kind of communication to the rest of cellular machinery that then allows for other processes involved in for instance, the hallmarks of cancer to be established. But with each of these, I mean, I guess the one point of introducing each of these proposals is that there are, pretty substantial challenges to each of these hypotheses. For instance, the tumor microenvironment, you can take a single cell in isolation of all of its, environmental multicellular, completely independent of its multicellular context. That single cell, if you turn on an oncogene, it will initiate the Warburg effect. And so, even in like unicellular yeast growing in isolation, they will do the Warburg effect. Biosynthesis, and also the Warburg effect is thought to be, an early event that's an immediate consequence of oncogene activation and occurs independent of the rest of the tumor microenvironment being established. In the case of biosynthesis, so one glucose molecule comes in, two lactate molecules come out. And so within the chemical equation, there's no stoichiometry that supports biomass. It's not like one glucose comes in and 5 lipids come out and 1 lactate comes out. The mass has to be conserved. When the glucose comes in and it gets fermented, that means it's fermented. That means it's essentially secreted as a waste product. It's not actually being used for biosynthetic materials. And so that's been a major challenge in terms of reconciling how this property of glucose metabolism may confer biosynthetic functions. And then signaling is more along the lines of the specificity, is how do you have this bulk property within the cell containing specific aspects of signaling. Typically we have signaling, we have a specific phosphorylation site is, a kinase gets activated, you have a specific phosphorylation site, and so on. But nevertheless, I mean, there are observations, and this is just an example of an experiment we did where, along this theme of the epigenetics, where basically you can, if you just titrate the amount of glycolytic rate in cells using this compound 2DG, basically you can show that there's global changes in histone acetylation that occur in a dose-dependent way, in a kinetic-dependent way, and then if you do something like mass spectrometry, where you quantify this basically these are cells undergoing 3 different rates of glycolysis, and you can see that half of the acetylome, or essentially the acetylation sites that are measured on histones, are dynamically altered by just controlling the glucose uptake rate. Okay, so that was the biology of the Warburg effect. Now, the other aspect of this is, okay, so if it's something that's so commonly observed and possibly fundamental to the development of cancer, is there a way to actually target it and actually figure out ways to think about therapeutic strategies or ways in which we can actually affect glucose metabolism in a tumor? And so there are a lot of challenges to this. One is that full enzyme inhibition is very difficult to achieve with a drug from just basic pharmacological principles. Provides some pretty substantial challenges to drugging glycolytic enzymes. So basic glycolytic enzymes are very abundant. So we did some calculations a few years ago, and basically the numbers we came up with is that glycolysis enzymes are about 10% of the entire proteome. So because the concentrations are so high, then you have to actually get drugs into cells to reach concentrations that are much higher than we typically consider when we, for instance, drug a protein kinase. The other thing is toxicity. I mean, glucose metabolism is something that's it's evolutionarily conserved in the most primitive of organisms. Every cell in your body uses glucose to some extent, especially your brain and your muscle, your liver. And it's required for all organ functions. So that's a major consideration in terms of thinking about what kind of therapeutic might be allowed for in this Warburg effect. So, and the other issue is, the existence of biomarkers. Is that typically when a lot of the current ideas in cancer medicine, have been centered around having defined, for instance, genetic statuses that can signify the or that have predictive value in determining a response. So you have a mutation in an oncogene, you have a drug that targets that mutation, and then you have a cancer that is affected by that principle. And so the case of something like cancer metabolism, where the alteration is really some combination of both you know, a collection of many oncogenes as well as many different environmental factors, like, for instance, I'm going to talk about some dietary things later on that, you know, all converge onto, you know, this metabolic phenotype. And so then thinking about, you know, these simple aspects of, okay, so can we do a simple test that says this is the particular type of cancer that responds to this agent that affects glycolysis is difficult. So we started to look at this by just revisiting a concept that's been around in biochemistry textbooks for over 50 years, and it's the concept of how rate control is distributed in the process. Basically, this is glycolysis, and then this is you know, classic undergrad textbook, and basically it states that there are 3 rate-limiting steps of glycolysis: hexokinase, phosphofructokinase, pyruvate kinase. And the basic idea of what's meant by rate control is that if you change those enzymes, that you exert a larger influence over the overall flow in the pathway. And so large rate control is when if you have a faucet, if you turn it a very small amount and a lot of water comes out, that's large rate control. If you have to turn it a lot and a very little amount of water comes out, that's small rate control. And it's generally thought that these 3 enzymes exert more control of the pathway. And so, so a few years ago, we published this paper in ELEC where we just did some computer simulations of rate control in glycolysis. And we basically had a model of glycolysis and then simulated all the kinetics of the pathway and did it both in the context of cells undergoing oxidative metabolism and the Warburg effect. And I think the major thing we saw in this model, what we were looking for was differences in rate control measured by this thing called the flux control coefficient. But we were looking for differences in the Warburg effect versus oxidative metabolism. And the major thing we saw is that in the context of the Warburg effect, this enzyme in glycolysis, GAPDH, glyceraldehyde phosphate dehydrogenase, was exerting more control over the pathway in conditions where cells carried out the Warburg effect, while other steps, for instance, PFK, was exerting flux control in both oxidative and Warburg effect. It was GAPDH that was not exerting control over the pathway in oxidative situations, but exerting control of the pathway in Warburg effect setting. So, so the next thing we did, so that at least gave us some hypothesis to start thinking about, you know, whether we could use that information to target cancer-specific glycolysis. And so first thing we did was try to look for a GAPDH inhibitor. And so we screened a series of several compounds that were reported in the literature to be GAPDH inhibitors. And I guess long story short, from these profiles, we measure a few hundred things in a dose-dependent manner in metabolism, and we look for one that produced a signature that's most consistent with specific changes in glycolysis. And this compound, kininic acid, which I'll introduce, was the one that basically scored the highest in terms of producing effects on metabolism that are most consistent with affecting glycolysis. And so this is just a zoom in on the glycolytic pathway as we treat with kininic acid. You can see that there's an accumulation of metabolites upstream of where the enzyme is, suggesting that at least there's an on-target effect here. So what is kininic acid? Kininic acid, turns out there's a really interesting, long-standing literature on this thing where basically it was identified in a screen for cholesterol synthesis inhibitors in the early 80s. It was isolated by Akira Endo. I actually didn't know who Akira Endo was, and then when we started getting interested in this compound, I was just looking him up on Wikipedia. I realized that half the pharmaceutical industry was working from compounds that he discovered from these screens from natural products in the Japanese islands that inhibited cholesterol synthesis. It turns out he discovered statins while he discovered this kininic acid, and so this one there wasn't a lot of literature on it, but it basically was something that was banked for a while. And the idea is that it's a natural product that lives in that is produced from a fungus that lives in sweet potatoes. And the idea is that when this fungus senses the presence of other fungi in the environment that may compete for the sugar resources in the fungus, it secretes this kiningic acid that has an antibiotic activity against anaerobic behavior, anaerobic bacteria. And this fungus, when it does secrete this compound with the intention of it being an antibiotic, the fungus simultaneously expresses a resistant allele of this GAPDH. And then there's been some biochemistry in the past that has shown that it's a nanomolar inhibitor of the human GAPDH. And so, so far we basically showed that we could do some metabolomics and show that it's hitting glycolysis, but how do we show that it's actually specifically targeting glycolysis? And so Maria, graduate student who was working on this, basically cloned the fungus version of the gene, expressed it in human cells, and you could show that the cells are completely resistant to inhibition with this compound. When it expresses the fungus version of the gene, suggesting that the cytotoxic effects of the compound are going through the GAPDH, or at least they require the GAPDH. And so this is just shown looking at metabolic pathways here. Basically, this is a, if we have empty vector versus this resistant allele, you can see that all kinds of stuff in glycolysis changes at a low dose of this compound, whereas if you put the resistant allele in and just profile every like hundreds of metabolites that happen in cells, like nothing changes. So then we quickly looked for what might determine the sensitivity of this kiningic acid. It was a collaboration with NCI where we screened it across a set of 60 cell lines at a 10 micromolar dose, and it turns out there's a whole spectrum of sensitivities to the compound. And if we looked for a lot of different signatures of what might predict the effects of the compound or the sensitivity of the compound. Looked at tissue type, presence of driver mutations, protein levels, RNA signatures, and so on. And it turned out, I guess, long story short, I'm going through this quickly as I realized I talked a little too much in the introduction, but I guess the long story short here is that when we scan all these potential molecular variables that may predict the effects of this compound, basically what we found is that the only thing we could observe that predicted the sensitivity of this compound was essentially the lactate secretion and the glucose consumption. That's shown here. What's shown here is on the plot on the x-axis is the the GI50 of the compound, and the y-axis is the flux that happened to be available for each of these cell lines. So basically, this is something where, at least in cell culture, if I said, okay, here's how much Warburg effect this cell is doing, I can tell you whether this drug may be effective or not. And so then we looked at this in animals, and this is the orthotopic xenograft model of breast cancer. You can show that at doses at which it achieves some efficacy. This is just showing tumor growth after a couple of weeks. So we can look at metabolomic. We can look at bulk parameters like body weight, blood cell counts, and so on. But but the other thing we do is we can profile metabolism in metabolic organs, something like muscle, and show that in these concentrations in which this compound is killing tumors and affecting tumor cell metabolism, it has no effects on metabolism in certain metabolic organs. This is an example of muscle. We also do it in liver and brain and so on. Okay, so just to summarize this, basically we had this quantitative model of differences in rate control and glycolysis, and that led us to think about GAPDH as an inhibitor that may have some selectivity over normal glucose metabolism, and basically through looking at panels of cell lines and certain aspects of mouse tumor models, that we could at least validate that this compound appeared to have some selectivity. Okay, so next part of the talk, and related to glycolysis, and this is how I got interested in epigenetics and this one-carbon metabolism, is that there's a side route pathway in glycolysis. Basically, glucose comes into cells. This is glucose to lactate, and it turns out when I was a postdoc in Lou Cantley's lab, I came there with the idea of setting up these metabolomics approaches, and that just once the technology was ready to start looking at things, we just screened a bunch of cell lines or profiled a bunch of things, and I guess one of the surprising observations was that there was a subset of cancer cells that were diverting their glucose out of glycolysis into this pathway shown here. And basically what this pathway is that there's an intermediate in glycolysis, 3-phosphoglycerate. It gets oxidized, transaminated, dephosphorylated to make serine, and then serine essentially donates its side chain into what is called 1-carbon metabolism, and then that gets accepted on folates and other amino acids which are introduced. And basically that side chain moves around and then outputs, as I mentioned earlier, different biological functions. And so the enzyme that initiates this committed step of this pathway is called phosphoglycerate dehydrogenase, which it's a dehydrogenase because basically it oxidizes the glycerate to make the intermediate that initiates the committed step of this pathway. So at the time this was discovered, I was at Boston at the time, and the Broad Institute was putting together collections of genomics data looking at copy number variations in human cancers, and so it turned out that on chromosome 1, there's a region of focal copy number alteration for which there was no known oncogene, and at the center of the region of copy number variation was this gene PHGH. And so this gave us some context in which suggested that this might provide some example of some tumors that have selected for the conversion of glucose to serine. And then we did some characterization of this. This is a ductal morphogenesis assay where you can express this phosphoglycerate dehydrogenase, and it disrupts acinar morphogenesis and prevents apoptosis in the interior of the gland. This is a model for mammary gland development, suggesting that this gene had some properties that were consistent with it being a cancer gene. So it is a druggable gene, and so this is something we've been exploring. And it turns out the catalytic site is very difficult to drug due to the fact that it binds to, well, you got a 3-phosphoglycerate, which is a very small molecule, and then you got NAD, which is a very big molecule, and both of those things are generally hard to drug. And so it turns out that we're able to identify some allosteric binding sites in the protein and make drugs around those binding sites, and that is something we published, and we're exploring this in therapeutic context now. And so, so downstream of this pathway, I mentioned that you have chemotherapies interacting on it, you have other nutrients coming in, and then you have all kinds of different pathways. And this is kind of when I began my independent research career, this is essentially what we built a lot of our research program on, was this, looking at functions of this pathway. And so I'll focus on one of those for the rest of the talk. And so this is one part of 1-carbon metabolism, the methionine cycle, where the basic idea of this methionine cycle is you have these nutrients come in. Nutrients came in the form of that serine, but also from this methionine intake. And then it's this 4-member cycle where basically methionine is converted to S-adenosylmethionine, then donates a methyl group to S-adenosine homocysteine, makes homocysteine, and then a methyl comes back onto it. And so basically methyl comes in, methyl comes out. And so this methyl group, something that was really intriguing, at the time we discovered this, in terms of just thinking about what kind of functions it might have in cancer. Essentially this methyl group was providing the substrate for things like histones and DNA. It's kind of shown, basically this is just a review we wrote recently where basically showing the different substrates in metabolism that were affecting chromatin, and basically this is that, for instance, histone methylation is involving the substrate, this S-adenosylmethionine. And then thinking about it in the context of cancer, classically this has been one of the one of the paradigms of cell transformation in cancer development, where you have a normal physiological process such as like a wound healing or immune response, where a growth factor is sensed, and then that growth factor initiates a signaling cascade. Signaling cascade then turns on, for instance, immediate early genes that then initiate a response. And so as cancer evolves, mutations that essentially uncouple the growth factor to the gene transcription are acquired in the tumor, and then those genes that get mutated provide a selective advantage to the growth of the cancer cell. So in the case of metabolism, we actually don't see a lot of mutations. I mean, there is this IDH1 gene, and then this phosphoglycerate dehydrogenase, which was less commonly observed. But what we've been finding the last 10 years in cancer as a result of the whole world of genomic sequencing and cancer genome atlas, and just the fact that sequencing a whole genome costs $1,000 in 2017, and it can be done essentially infinitely in cancer settings, is that the classes of genes that are recurrently mutated, that we're finding a lot more mutations in, are these chromatin modifying genes. Things like methyltransferases, things like chromatin remodeling complexes, demethylases, and so on. So possibly something that may be going on here is that, in the same way we have this analogy where you uncouple the growth factor from the transcription when you get the mutations, whether you actually uncouple the metabolism from whatever's happening to the chromatin by conferring these mutations, and potentially that's where metabolism is possibly at least involved in the process of cancer. So, I mean, there are biochemical principles in terms of how this may happen. The basic idea is shown on this this Michaelis-Menten curve here, where the basic idea is that post-translational modifications require a metabolic substrate and an enzyme. And in the case of protein kinases, the Km, the Michaelis constant of that enzyme, is something on the order of micromolar, whereas the concentration of the substrate, like ATP, is on the order of millimolar. So there's a 1,000-fold difference in the concentration of the substrate and the Km. So you never get situations where cells don't have enough ATP in order to carry out the biochemistry involving protein kinase reactions. And so that's in sharp contrast with these methyltransferases and acetyltransferases in cells, where it turns out that the substrate, this S-adenosylmethionine, is right on the order of the concentration, of the Km of the enzyme. And that's just, for instance, shown here in this table, where we just did a quick literature survey of just all the in vitro biochemical parameters that could be measured for different methyltransferases, acetyltransferases, and so on. And basically what we found is that these substrates were, the concentration of these substrates were fluctuating right around the values of the Kns of the enzyme, suggesting that this is something where if you change metabolism, you have a huge effect on the rates of these enzymes, which then has a huge effect on the levels of the modifications, which then could have all kinds of aspects of changes in genomic architecture. And then, this is just one example where we did this experiment where we just measured methionine concentrations in humans, in fasting serum, and basically saw that this was the range of methionine concentrations, about 3 to 35 micromolar. And so normal cell culture contains about 100 micromolar of methionine. And so we did an experiment where we just said, what happens if you just go from 100 micromolar methionine and reduce it down to the lower limit of what's observed in humans, like 3 micromolar? And what we found is that you could see major changes in certain histone marks. This is one example of a histone mark for which there's a lot of biology around it that had a fairly substantial dynamic response. This is something we published a couple years ago, and just the summary of that is that basically what we did in that paper was kind of show quantitatively with the kinetics and the values of the Km that basically this was something that was sensed by the availability of methionine, was this histone mark, that it was very dynamic, reversible, it altered histone methylation, and then we also showed that it could affect, could be affected by diet. So I realize I'm running a little bit out of time, so I'll be quick about a lot of this stuff. But so this H3K4 trimethylation, there's a lot of literature done by other groups, for instance, Anne Barnet's lab, Wei Li's lab, Baylor basically showed that there's a lot of information encoded in how this mark is deposited across the genome. So we looked at this a little bit, and then did quantitative ChIP-seq in the presence of methionine, high methionine, low methionine, under those conditions where we saw histone marks, but also did it in mice where we fed the mice low methionine diets or normal methionine diets, and collected, for instance, their livers. And basically there's cell type specific changes in the peaks. Basically there's information in the genomic architecture that's encoding what's happening to the changes of these peaks. It's just shown here. This is just a cancer cell. This is a normal physiological context. So there's a mouse eat a lower diet, and you can see that there's it's enriched in certain transcription factors that are involved in these processes. For instance, these are cancer transcription factors, and these are liver functioning transcription factors. And also the changes in the width of the peak is also what was predicting gene expression. So just to think about this in the context of cancer, okay, so I mentioned this dietary methionine. So basically there's a lot of literature around methionine having some, for unknown reasons, health benefits. Basic idea is that if you restrict the concentration of methionine in the diets of model organisms, this is conserved all the way to yeast, but these are just examples that are found in rodents where a normal laboratory animal eating a normal diet that is administered contains about 0.85% methionine. So if you restrict it to 0.15% methionine, there's a lot of phenotypes. One, it actually extends the lifespan of the animal. Two, it will induce weight loss, and it's thought that this weight loss is through changes in glucose metabolism and metabolic parameters. Basically, the mice have better insulin sensitivity as a result of this diet. So I guess what we wanted to think about was whether this diet had any effects in cancer settings. So the first thing we did was just see how controlled was this particular diet. And so still we have something where, where we're looking at just a mouse eating a diet that's just altered in that's isocaloric, but just altered in one specific amino acid. And so, I mean, the first thing we want to know is whether this is something that could actually be controlled in a physiological context. And so we just profiled the dynamics or the kinetics of this diet. From day 0, the mice go on the diet, and then we look through tail bleeding, what's actually happening over time. And you can see that there's actually very specific changes in methionine metabolism happening as these diets. So I mean, because the alternative hypothesis would be that there's so many physiological ways to adapt to something like a change in the diet that consistent metabolic changes may not be apparent. But apparently in this dietary situation of just restricting the concentration of one amino acid, you can see that there's localized effects on methionine metabolism. Something I briefly mentioned, that this is also sufficient to change levels of histone methylation. These are livers from mice that have been eating this diet for about 3 months. Okay, so yeah, and then in the context of cancer growth, we look to just apply this model or this dietary system to a panel of different kind of cancer models. This is one spectrum of the type of cancer model that's of consideration in preclinical settings. These are patient-derived models where the tumors have been explanted from patients and then immediately placed into mice, and these are early passage through the mice, and these are just 2 examples of RAS-driven colorectal tumors that seem to respond by this change in the diet. This is a chemo-resistant model where if you couple the chemo resistance, this is normally in the presence of the 5-FU, which also targets another aspect of 1-carbon metabolism. The diet then appears to interact with it and affect tumor growth. This is another model. This is an autochthonous tumor model that's driven by, it's also driven by KRAS as well as a loss of P53, and then the Cre is delivered to the muscle. It forms a sarcoma, and this is a model of radiation resistance. And you can basically see that if you couple, so the diet doesn't do anything in this model by itself. And then if you combine it, if you just irradiate the mouse, give it focal radiation of fairly high dose, it has some effect on the, reducing the tumor growth. But then if you couple the radiation to the diet, then you can get, a synergistic outcome with the diet. And so So something to note is that the first time I mentioned before, it was a chemotherapy targeting 1-carbon metabolism. And this one's radiation, which also maybe indirectly affects 1-carbon metabolism by influencing by causing DNA damage and requiring more nucleotides to be synthesized. So in both cases, you have something that's molecularly coupled to the diet in which there's an effect that propagates. And so, yeah, just the last thing to just quickly mention some of the stuff we're doing in humans. So it turns out this is actually something you can functionally do in a human being, or may possibly be able to do. And so this is basically what a methionine-restricted diet looks like in humans. This is a pilot study of about 6 volunteers that basically volunteered to eat something like this for about 3 weeks. And then, we do the metabolomics, and basically you can see that we're basically targeting 1-carbon metabolism, like, with the diet. And you can see actually localized effects with the diet by just designing something that, appears to have isocaloric, like macronutrient-defined, compositions, but just missing one amino acid, or at least lower in one amino acid. And you can see, that you can basically suppress, like, the 1-carbon metabolism and the methionine cycle in those humans. Okay, so yeah, just to summarize this point, I mean, this, I didn't get into a lot of the epigenomics. I realized I just had too much stuff in here, but basically this methionine cycle seems to be something that physiologically interacts with chromatin. It's just normal responses where you have variation in what's happening in 1-carbon metabolism that is directly coupled to changes in changes in chromatin. This is just a model system. We've been using this H3K4 trimethylation because it's something that's very dynamic and responsive, and it has gene programs associated with it. It appears, as I mentioned, it appears to occur in physiology, and it may have cancer. It does produce cancer phenotypes in different preclinical settings, and then it appears to be actually fairly specific to actually affecting 1-carbon metabolism in those settings. So just lastly, I want to thank a few people who really did the work for this. I mean, especially Maria did the, she's a senior grad student in the lab. She did a lot of that Warburg effect targeting work that I presented earlier. Bella's a postdoc trained in physiology. She did a lot of the diet stuff, and Zoe is a computational biologist who did a lot of the ChIP-seq and metabolomics analysis that I presented. With that, happy to take any questions.
Just a comment.
Yeah.
It's sort of a historical note. 30 to 35 years ago, there was 2 faculty here. One, Gabriel Fernandez, working with a guy named Edmund Eunice, who was an immunologist. Mm-hmm. And they did some phenomenal work about calorie restriction. Mm-hmm. And they never figured out what was going on, but if you cut the calories 25% so unless they put tumors in, A, they live much longer, B, the tumors are much smaller, and whatever. Obviously it wasn't as sophisticated as what you have shown, but how has the caloric intake field relative to cancer, and has any of those studies carried on?
Yeah, I guess the short answer is it's an active area. I mean, and it's something where we're like, there's caloric intake, there's how you couple fasting versus fed with drug intake. That also has things. I mean, what I was showing here was that there could be, generally speaking, I guess the first variable is whether the total amount of calories affects an outcome. The next variable is whether it's the macronutrient composition, whether it's the amount of carbohydrates versus lipids, and then there's a whole nother level of potential detail here where you're really like same macronutrients, same amount of calories, but you just change one dietary variable and it seems to have outcomes. And then the what we're all kind of working through are what are the mechanisms and why. Like what's going on in each of these situations is probably just as complex as the genome. It's just something that, I mean, one thing we like to do with this is just couple it all to this metabolite profiling, metabolomics, and I think at the very least, and as far as we want to go with a specific conclusion in terms of mechanisms, we can show that this methionine actually seems to specifically localize and target around a specific metabolic pathway, and that was something I think that we wanted to at least emphasize with this study. But then, and then there's a whole field that is looking for calorie restriction mimetics and what kind of processes might phenocopy the effects of calorie restriction. It's fairly well established that that does have health outcomes. It's also fairly well established that it's not something people want to do.
No.
And so
History proves that every day.
Yeah, and so, so then there's a whole world of like figuring out what you can do, and yeah. Oh, sorry, it's just.
Question?
So I was wondering, you touched on this a little bit, if you could expand a little bit about the connectivity between the metabolome and its output and epigenome in general. And
yeah.
You mentioned the notion that there may be tumor contexts, for example, where that relationship becomes modified based on mutations in the genome or oncogenes, etc. So is that relationship dynamic in nature? And how tight is that relationship in general? Because let me just give you a context.
Yeah.
There's obviously a lot of discussion, for example, about post-translational modifications, including acetylation, and how much of this is just sort of the sort of trash heap of the metabolome that's actually influencing enzymatic machinery, that's actually specifically guided versus that which is targeted and regulating in nature. And so what is your take on that, and how dynamic is this in nature?
Yeah, so, okay, so in terms of the epigenome and modifications, I think you're absolutely right in which there's a lot more modifications with no known function than there are with function. That being said, I mean, I guess the major, I guess there's two things that epigenetics has going for it right now that's kind of putting it into major focus and biomedical research. One is that in cancer biology, a lot of the new mutations that we're discovering function to influence these modifications. Like, for instance, something we've been starting to work on, so there's certain pediatric brain cancers where it's actually mutations in specific residues of histones that don't allow for the modification to be conferred that is the initiating event of the cancer. So that's one thing. And then the other thing that it has going for at the moment is that the therapeutics do seem to be, I mean, they have activity, meaning that you can block enzymes that put on these modifications. You have enzymes for which there is no known function of these enzymes other than to affect an epigenomic modification, and if you target that enzyme, then you get effects on the tumor. So, I put up some model earlier where I said maybe where metabolism is coming in terms of driving the development of like pathological conditions like cancer is when, if you have normal coupling of metabolism to the chromatin and then you have mutations which function to co-opt this coupling, and how established that is at the moment, I wouldn't say, but at least it's just some idea that, at least kind of comes from, some of the more established models we have of cancer development, like when, for instance, like growth factor signaling is dysregulated by mutational events that, at least like those kinds of models at least appear to, in some cases, to fit that dynamic, but the extent that it happens overall in biology is that part's not clear, and cancer is not clear either.
In your methionine restriction study, you observed decrease in histone methylation.
Yeah.
How about DNA methylation? It might be even more relevant to.
Yeah, DNA methylation is more complicated. So the other thing that happens is, because there's a lot of things. Okay, so you alter methionine metabolism. You've got direct effect, like. So in a cell culture model, like we can work out the kinetics and we can look at short-term effects and do more quantitative analysis of how the metabolism interacts with the chromatin. In the physiological situation where you're putting an animal on a diet and letting it go for a long term, one thing that happens is that there's transcriptional programs that compensate for the nutritional alteration, but also you also have direct effects on the chromatin. So when we do methylation, we actually see a global increase and then changes in specific sites, and the increases, and we can also see that there's a lot of methyltransferases that get upregulated and suggest that there's some response happening, but the final outcome of that response would then really depend on the context of the situation.
Yeah, we'll ask just
Yeah.
one question. We have that's room.
Oh.
All right, well, thank you very much. You know, about that, the GAP-DH data,
Mhm.
that you showed that the cells dependent on the Warburg effect are dependent on GAP-DH,
Mhm.
essentially. Do you see that those cells upregulate GAP-DH and gene expression?
Oh, so yeah, so that's, okay, so basically this is entirely a property of the whole metabolic pathway. So basically you have the same protein expression, you have the same enzyme activity of GAP-DH. It's just that if you make a change to GAP-DH, then the rate at which the whole pathway flows, it has to do with how the thermodynamics is set up in the pathway and the different, you know, in Warburg versus in fermentation versus oxidative conditions. But basically it's a property of the whole network where
But you look for, like, you haven't seen evidence that that cancer cells actually upregulate GAP-DH.
oh, so they don't upregulate GAP-DH, yes. So they don't upregulate GAP-DH. What they do is they upregulate the Warburg effect. The Warburg effect is turned on through all kinds of different mechanisms. Like the simplest mechanism which a Warburg effect gets established is you have oncogene. Oncogene activates these two transcription factors, MYC and HIF, and those things hit about 10 different, there's about 10 different enzymes in glycolysis that all come up together. And that'll give you a Warburg effect. That's a very easy way to get the Warburg effect. What we're saying here is that once you have a Warburg effect, then do the enzymes in and of themselves have different properties? And meaning that, you know, they can have the same expression, they can have the same activity, the same intrinsic activity of the enzyme, but it's just that if you affect that activity, then does it actually affect the whole pathway or not? And so basically what we're showing here is that basically what you can do is like a 10% change in the GAP-DH activity has a big effect on overall glucose metabolism when you have a Warburg effect. But if you do a 10% change in the activity when you have normal conditions of glucose oxidation, then that has no effect. And that's the idea.
I did single cell transcriptional profiling of leukemia stem cells, and I do see differential expression of GAP-DH.
Oh,
and that's
okay.
why this is sort of fascinating.
Yeah.
And these leukemia cells have, well, I don't know actually, anyway.
Yeah.
So.
Yeah, I'll have to
Oh.
say you can just come out and ask him. Is that okay? Because we have to let the room go.
Okay.
Thanks, yeah.
Okay, thanks everyone.